Good day. I'm Professor Richard Burt, and I developed the field or pioneered the field of stem cell transplants, that is, blood or hematopoietic stem cell transplants for autoimmune diseases. And I used to run an immunology lab. So I want to talk today about immunity, that is, the balance between cancer, infections, and autoimmunity, and going from bench to bedside and back, and how that has improved in humanity and gives you an understanding of why we're doing what we do, not just me, but other people. So I think the first and most important point about immunity is to understand there are two types of immunity: that is innate, and that's directed against many types of pathogens. It's not specific for a certain type of infection, and adaptive, and that is specific for a unique pathogen. So life protects from pathogens by using two immune systems. Innate requires no prior infection, and you just have this broad immunity. And adaptive, which needs a prior infection to prime or make your immune system able to respond to a specific pathogen. And this occurs in both bacteria and in humans. So there's a war between bacteria and viruses, and bacteria have the innate immunity and they have adaptive immunity. And that adaptive immunity in bacteria is called CRISPR or CRISPR-Cas, and it gives you a specific defense for a specific virus that's trying to attack the bacteria. Humans also have innate immunity, which is broad to many types of infections, and an adaptive immunity that is learned and becomes specific for a unique type of infection. And that adaptive immunity in humans is T and B cells, which is different than the CRISPR adaptive immunity in bacteria. So first, I'll talk about bacteria. Since both bacteria and humans have both innate and adaptive immunity. So bacterial has innate immunity, which are proteins that block virus binding, entry, or replication. And it does it for any type of protein or a broad different types of proteins. And bacteria contain restriction endonucleases that are enzymes that cut viral proteins if they get into the bacteria. And the bacteria themselves tend to methylate their DNA to prevent the restriction endonucleases from cutting the bacteria's own DNA. Well, these restriction endonucleases tend to cut in what's called palindromic sequences. Palindromic means the same way forward as backwards, like for the word radar, spelled forward or backwards, it's the same. So in orange, you see GAATTC in one strand. In the complementary strand, it goes backwards the same way, reading on the bottom from right to left, GAATTC. So when it sees these palindromic sequences, it'll make a cut. So with the discovery of restriction exonucleases in bacteria that are designed to protect the bacteria from an infection, once we discovered those restriction endonucleases, that led to cutting and ligating or placing genes into plasmids. So what is a plasmid? So plasmids are extra chromosomal circular DNA that's also found in bacteria. So bacteria have chromosomal DNA. It's not in a nucleus, it's just chromosomal DNA, but it also has small circular extrachromal DNA called plasmids. And if you take the plasmid that you can get from bacteria, insert a gene in the plasmid that you want expressed and put it back in the bacteria, and that's done by electroporation, really just electricity to open holes in the bacteria, or liposuction, that is, it's kind of surrounded by a lipid membrane that binds to the bacterial lipid membrane to enter. Once it's in there, it'll make the protein you want from that gene. A lot of cancer biologics are now made that way. So this discovery of the innate immunity of restriction endonucleases allowed us to produce a lot of different types of proteins to be used as drugs for human beings and to manipulate DNA in plasmids to insert genes. So that was innate immunity in bacteria. What about adaptive immunity in bacteria? That is, bacteria will develop a response to a specific virus. And how does it does that? It uses a system called CRISPR. So CRISPR is an abbreviation for clustered regular interspaced short palindromic repeats. So within the chromosomal DNA of bacteria, there's a CRISPR array that contains these palindromic repeats, these kind of squares that are labeled as R, the green R's. And between those are the S, or spaces, where segments of viral DNA is placed. So R is palindromic repeats, S is spacer, where a piece of a phage or viral DNA is put into the chromosome of the bacteria. So how it does that is it uses Cas, which stands for CRISPR-associated proteins, that cut a small piece of this viral DNA. There's the phage virus injecting its DNA into the bacteria, cuts it, killing the virus, but upon cutting it, it inserts it into the chromosomal viral DNA of the bacteria in an area called the DNA CRISPR array. So the CRISPR-Cas complex preserves a memory of the past viral infection in the bacteria. So the question is, how does putting viral DNA into a bacteria chromosome serve to develop adaptive immunity to kill that specific virus the next time it's infected by it? It's an adaptive immunity specific for that virus, but putting a piece of that viral DNA into the chromosome bacteria, how does that work? Well, the CRISPR system does that. And another acronym, not the formal one, but one to kind of remember, is capture, RNA transcription, interference, surveillance, phage destruction, and then repeat. So what happens is when the virus infects the bacteria, a piece of the DNA is cut and captured into the CRISPR array. Once it's in that array, the bacteria itself will transcribe that viral DNA into RNA called pre-CRISPR RNA. And then that pre-CRISPR RNA, which is a guide RNA, gets proteins bound to it, that is Cas proteins, CRISPR-associated proteins, to form the Cas CRISPR RNA complex. So what that complex does, it circulates around inside the bacteria as a surveillance complex searching for that virus in case it infects the bacteria. If it finds that virus, it destroys it, and it destroys it because that guide RNA from that saved piece of viral gene binds to the virus, and then the Cas proteins cut the virus and degrade it. So it gives specific immunity to that virus. So although bacteria use this CRISPR-Cas complex for adaptive, that is, specific immunity to a given virus it's seen before, the CRISPR-Cas complex, which is a guide RNA and like different types of Cas proteins, but in this case Cas9, is now used to target or precisely edit the human genome. Because if you want to edit it, you just have your guide RNA match the sequence of the gene you're interested in. It'll bind there, and the Cas proteins can cut it out, and then you can insert the gene you want. And you can insert it specifically where you want in a human a chromosome. And that is ongoing. So that was bacteria, and what, in brief, their innate and adaptive immunity, and how we've been able to harness that from bench to bedside to create a variety of human drugs and to start engineering cells, human and non-human, to express different genes or that you desire, and to put them in the right place in the chromosome. So there's not, carcinogenesis where it falls or activates a cancer gene or blocks a suppressor gene. So that was bacteria, but humans have two immune systems as well: an innate immunity, and those in red here, the red circle, is the innate immune cells, neutrophils, macrophages, dendritic cells, and an adaptive immunity, which are basically your T and B lymphocytes. Again, innate immunity, non-specific attack on whatever is infecting you. Adaptive immunity, it has to see it, and then you learn from that so it can specifically attack that fungus or bacteria or virus in the future. So human innate immunity, that's non-specific, and it attacks immediately. It doesn't need to learn. Innate immunity reacts fast. It's nonspecific, does not require prior exposure to the infectious agent. That's why it can react so fast. The first line of defense is your innate immunity, and it gives you that first week of survival for innate immunity to develop, because it takes about a week for innate immunity to develop, to learn what that particular infection is, and then to assist in defense against that pathogen. So your first line of defense that holds the line for you is innate immunity. So innate cells, or the innate immune cells killed by phagocytosis, that is eating what pathogen it finds, by a variety of proteins or glycoproteins like porphyrins, granzymes, complement, interferons, interleukins, fever itself, and inflammation from a variety of different prostaglandins, histamines, and bradykinin. That's all innate immunity. So innate immune cells, including neutrophils, macrophages, natural killer cells, those are all your innate immune cells. So to begin with innate immunity, we have to understand that the human body, the topology of the human body, we think when we go in our mouth, we go on the inside. We don't. The human body is a Mobius strip. The inside connects to the outside. And therefore, the innate immune system stops pathogens before they enter the body. It stops it in the inside, in our mouth, in our esophagus, in our small bowel, in our colon. It stops it before it can enter the body. All that area, our entire GI tract, reconnects to the outside of the body, whether you're going through the mouth or out the anus. It reconnects. So it's like a Mobius strip where you end up back on the outside. And so the innate immune system also protects the outside from pathogens getting inside your body. So outside the body, we have dry epithelium, that is skin. And this epithelium is a protective layer. We have sebaceous glands, and the sebaceous or sebum fluid is very acidic, which kills bacteria. Bacteria don't like acidity. And of course, if the sebum is really copious and can mix with dead cells and hair follicles, you get acne. We have sweat, and sweat, if you've ever tasted your own sweat, is salty, and bacteria don't like salt. And sweat also contains antimicrobial peptides, which is abbreviated AMP. Our tears contain salt, if you ever taste your own tear, and antimicrobial peptides, such as lysozymes, to kill bacteria, so that we don't get conjunctivitis or infections around our eyes. And the inside of the body, in this Mobius strip, you're not really in the body, you're still on the outside, but you go through the mouth on down to the, where our food would go. We have saliva in the mouth, which is loaded with antimicrobial peptides that non-specifically kill a variety of pathogens. That's why when you look at a dog, when it gets injured, it licks itself. It's transferring these antimicrobial peptides to the wound to kill bacteria. You have mucus as you go further down, either into your lung. When you have a cough, you're coughing up mucus, or go down the esophagus, you have mucus. And this mucus, or slime, if you will, has antimicrobial peptides and that kill and provide defense against pathogens, but it also prevents bacterial adherence to your cells, which they need to get through the cells to get inside your body. And then you have cilia, often in mucous membranes, that moves the mucus and bacteria outside the body. So if you get a upper respiratory tract infection, you start coughing, the cilia help move that mucus with bacteria in there. It turns yellow or green, and you cough it out. Your stomach is also very acidic, and that helps kill bacteria that you eat in food when it gets to the stomach. So innate immunity kills pathogens once they get inside the body. I just talked about how innate immunity in humans, mammals, whatever, prevents pathogens from getting in the body, whether you're eating them or whether they're on your skin or your eyes or wherever. It prevents it from getting in, but it also kills it once it gets in the body. And innate immunity includes all these things that causes inflammations like nitric oxide, histamine, bradykinin, and prostaglandins that are produced by these different innate immune cells like neutrophils, macrophages, so forth, mast cells. And what that gives is it dilates, makes the vessels more permeable, and it makes that area erythematous are red, warm, painful, and swollen. And that then we recognize as a sign of infection. But the reason that happens is that so cells can enter both innate and adaptive immune cells to fight the infection, in the tissue of your body. So cells of the innate immune system uh, release a whole group of proteins or glycoproteins to fight infections, including complement that can be activated to kill bacteria, interleukins and interferons that cause fever and also help activate the adaptive immune system. But fever inhibits bacteria and viruses from growing. It causes more damage to them than to our own bodies. It's a defense. And then it also, these cells release, again, a variety of antimicrobial peptides at the site of where the infection has penetrated inside your body. So besides all these proteins and glycoproteins, the cells themselves of the innate immune system eat this pathogen and also release a variety of antimicrobial peptides. This is done by neutrophils, which are kind of your first responders that get, the vessels open up, dilate, you get the erythema, the warmth, the neutrophils flood in there, initiate the attack. You have monocytes, which are wandering macrophages, and tissue macrophages that are stuck and named according to the tissue they're in, like histiocytes, Kupffer cells in the liver, microglia in the brain, so forth, that will attack and consume pathogens. And then most importantly, you have dendritic cells. And these are the best cells at activating adaptive immunity. So your innate immunity is your first line of defense. If it's overwhelmed, it will buy time, about a week usually, for activation of your adaptive immunity if it's never seen this pathogen before to fight it. And the main cell doing that activation is your dendritic cell. So, as a quote from Charles Janeway, who's the father of human immunology, a foreign antigen alone is insufficient to elicit the adaptive immune response. It requires co-stimulation from innate immunity. So you have two immune systems, innate and adaptive. Innate will just attack your pathogen. It's specific for the pathogen, and it attacks a broad range of pathogens. But you have adaptive immunity as well, which is specific for a given pathogen. It has to be learned, and how it learns, how your T or B cell learns to respond to a specific pathogen, it requires interaction with and co-stimulation from innate immunity. It's kind of like your first line of defense. Innate immunity is fighting the battle, but it talks to the adaptive immune system to educate it, to help it come as a backup, as the cavalry to fight that infection. So human adaptive immunity. In terms of adaptive immunity, we can discuss B cells, but there's not enough time. So I'm only going to discuss the T cell. And basically that's also because immunologists tend to work in very specific areas. And a bench immunologist will work only in T cells, or only B cells, or only dendritic cells, or one type of dendritic cell, for example, or one type of T cell. I ran a T cell lab. So adaptive immunity arises by DNA rearrangement of variable diversity in joining genes to form naive T lymphocyte with a T cell receptor that is unique for one type of infectious organism. Now, this is a random process, so it'll form a T cell receptor that can recognize and potentially damage self-epitopes, self-antigens, or self-tissue. So that's where you can get overlap or a derangement between an immune attack against a pathogen and attack against your own body. Because this rearrangement is a random process. So the naive lymphocyte, after that rearrangement, which occurs in the thymus, it's naive. It's not going to respond. It can bind to self or to pathogen, but it won't do anything. It's anergic unless it's co-stimulated by the innate immune system. So the innate immune system, dendritic cells, sees a pathogen, it engulfs it, it's trying to destroy it, presents pieces of it to a T cell with co-stimulation that activates the T cell to the pathogen. It can go awry and activate it to a self-epitope and cause autoimmune disease as well. I'll talk about that a little later. So the adaptive immune T cells can not only be an effector cell, that is a CD4 or CD8 lymphocytes that attack a pathogen and cause inflammation. It can be a regulatory T cell, a lymphocyte that decreases inflammation. So your T cells, again, just like you have a dual immune system, innate and adaptive has to be activated by innate, you have two types of large types of T cells. One is regulatory to suppress inflammation, and one is effector to start it. And that's important because once you start an attack, you need to shut it off. So, as kind of an aside, going back to CRISPR-Cas9, that is now being used to knock out variable joining diversity genes that are a unique T cell receptor and insert the variable joining diversity gene you want for an engineered T cell receptor. So you have an engineered T lymphocyte that you can use in immunotherapy to attack a tumor. Those are called CAR T cells. All right, T regulatory cells. These are lymphocytes that decrease inflammation, and Tregs may be either CD4 or CD8 positive, but they're also FOXP3 positive. So a natural Tregs are selected or developed in the thymus, and they have a very high avidity, that is affinity, for the MHC self-peptide complex. And the reason for that is by that high avidity to identify self in a MHC molecule, major histocompatibility complex molecule, and because it's a Treg, it can help prevent autoimmune diseases. Induced Tregs are generated in the peripheral mucosa of your respiratory tract or your gut, and they help prevent environmental allergies, so like asthma and things like that. So Tregs are low in autoimmune diseases like lupus. And in some autoimmune diseases like multiple sclerosis, they're not low, but the T effector cells that can cause disease and destroy myelin are resistant to the suppressive effects of Tregs. So these Tregs can become abnormal in autoimmune disorders. So again, I want to look here so you understand there are natural Tregs and induced Tregs that help suppress immunity. The natural Tregs, called N-Tregs, are made in the thymus to minimize autoimmunity, and they generally have a pretty strong avidity or affinity for self-epitope to prevent your other T cells, your effector T cells, from causing damage to self. Your induced Tregs are made in mucosal tissues like the respiratory tract or gut. And in the gut, they prevent food allergies. And in the respiratory tract, they prevent airborne allergens like asthma or hay fever or whatever. So again, how this happens in the induced, you have in the periphery of dendritic cells that presents these allergens along with TGF beta and retinoic acid RA there to a naive CD4 Treg, and then that gives you an induced Treg. So Tregs markedly increase after hematopoietic stem cell transplantation for autoimmune diseases. And I said I had pioneered the use of hematopoietic stem cell transplant for autoimmune diseases like multiple sclerosis, CIDP, Crohn's, neuromyelitis optica, and systemic sclerosis. Well, one of the ways that work is that the transplant causes a marked surge in Tregs. They increase afterwards. These help shut down inflammation. So I talked about Tregs. Now it turns out you also have these effectors, CD4, CD8 T cells, that do the opposite. They cause inflammation. They attack and kill pathogens and tumors, and they are made in the thymus. So when you think of CD4 CD8s, I like to think of CD8 cells as cold killer cells. They're your assassins. They're also called cytotoxic T lymphocytes. They're effector cells that kill a pathogen or a tumor cell, and they often do it by a combination of fast ligand, or granzymes that induce apoptosis. And then I like to think of the CD4 cells, your teacher cell. The CD4 lymphocyte is a helper cell or a teacher cell that releases cytokines to help instruct or teach other immune cells, including CD8 and innate immune cells, macrophages, other cells, B cells, what to do. So CD4 and CD8 cells are produced in the thymus, these effector cells, but they are deleted in the thymus if they bind really strongly to self. So if they bind too strong to self peptide and MHC molecule, they're deleted, they die. That way, they're not released into the periphery where they could cause an autoimmune disease. If they bind with high avidity, but not super strong avidity, they'll become a Treg to help shut down attack on self. Really strong, they're just deleted. So your CD4, CD8 effector cells are selected for low to moderate avidity to self. So T cells must recognize self with moderate avidity to survive and exit the thymus, where if they're instructed by a dendritic cell and they recognize this self epitope on a pathogen, they will join in the attack. So autoimmune cells are normal, but autoimmune disease is pathogenic. That's an important point when you do stem cell transplant for autoimmune disease, because the technology came out of oncology where they do stem cell transplant for cancer, leukemias, lymphomas, so forth, where the concept is cancer, malignant cells, evil, kill it all. You can't kill all your autoimmune cells. They are normal. The lack of cooperation and interaction between them and lack of the appropriate balance is what causes autoimmune disease. All you want to do is reset that balance. So how does the thymus make CD4, CD8, in this case, CD8 cytotoxic T cells? Well, it starts from the hematopoietic stem cell in the bone marrow, and it has a surface antigen, CD34 or CD33. Those are the antigens, the hematopoietic stem cell. And in the bone marrow becomes an early pan T cells, and those early pan T cells have CD2, CD5, CD7 on their surface. It then circulates into the thymus, where it becomes double negative CD3 positive cell. CD3 is a marker for a T cell. CD3 is not on the early pan T cell lineage. That's CD2, CD5, CD7. In the thymus, those are changed to CD3, which is present on T cells. But it's called double negative because it doesn't have a CD4 or CD8 marker. And then it becomes double positive, where it has a T cell receptor. That's the random variable joining diversity gene comes together to make a T cell receptor to recognize about a 10 to 12 amino acid sequence of a pathogen or self tissue. And at that point, it's both CD4 and CD8 positive, as well as CD3, which is a T cell marker. But once it exits the thymus, it's a single positive CD8 cell or a single positive CD4 helper cell. In this case, I showed the single positive CD8 cell as cytotoxic T lymphocytes, contains CD3, pan marker for a T cell, CD8 for being a cytotoxic T cell, and the T cell receptor, that recombination of variable joining and diversity gene. So these cells, when they're made in the thymus, they're naive. They're not going to react to an antigen or an epitope. In the periphery, these post-thymic single positive CD4 or CD8 cells, which are naive, become activated. So they're not initially effective against an infection, and they're also not initially dangerous to self. They're anergic. They have to be activated by a dendritic cell, which is part of your innate immune system, to attack that pathogen. In the periphery, the single positive CD4 or CD8 cells can become activated to a foreign antigen, a pathogen, or to self-antigen in peripheral lymphoid tissue when presented with antigen and co-stimulation. And again, this is the interplay between this dual immune system and how it's very important in protecting us from pathogens, but it can also go awry and give an autoimmune disorder. It's also important, by the way, in terms of killing a tumor cell. So what I want to show is the peripheral activation of a CD8 T cell or cytotoxic T cells. And so in the periphery, this naive single positive CD8 T cell will find a dendritic cell and become activated. And then it undergoes this clonal expansion. And then these short-lived effector CD8 cells will kill a target tissue. And that target tissue is destroyed. And that target tissue is a cancer or an infected cell, a cell infected by a virus or whatever. And it will kill it and get rid of it. But it also, besides making these short-lived effector cells that will kill the pathogen or the tumor cell, it leaves behind adaptive memory cells that have that specific a T cell receptor of a variable joining diversity gene rearranged that it now exists and can be immediately activated to infection. So you don't have that week delay before the adaptive immunity is there, and it's specific to a particular pathogen. Now, these memory cells are given different names. I won't go into, T central memory, T effector memory, tissue resident memory, whatever. So CD8 T cell activation requires presentation of co-stimulation. So not just the antigen of the pathogen, or if it's confused, a self-antigen in the MHC a molecule, but co-stimulation by other molecules such as CD8, CD20 on antigen-presenting cells to stimulate your CD4 cells or CD8 cells to kill tumor and elicit infections and eliminate infections, excuse me. So here I show the MHC antigen so that the T cell receptor recognizes a specific MHC antigen complex. And normally, if it recognizes, it's not going to do anything. It's got to be co-stimulated to become active. And the antigen presenting cell, which is innate immune cells, recognize this pathogen as dangerous. And so it provides that co-stimulation to the T cell, whether it's CD4 or CD8, that can then participate in stopping this pathogen. So, of interest, low CD8 cytotoxic lymphocytes, or a CD4 to CD8 ratio in your blood less than 0.8, increases your risk of getting a lymphoma, sarcoma, colorectal disease, lung cancer, etc. It increases the risk of cancer. Nobody really follows those, but in the papers that have studied it, it does increase your risk. So chimeric antigen receptor T cells, which are now called CAR T cells, that's an abbreviation of chimeric antigen receptor T cells, are used to treat cancer, especially B cell lymphomas or ALL that are B cell driven, and they target the B cell, this CD20 molecule in the B cell. But because some autoimmune diseases like lupus are predominantly B cell mediated, it targets that B cell epitope being used to treat lupus as well. Those CAR T cell receptors not only target a specific antigen or epitope, but they often include, when they're engineered, intracellular co-stimulatory domain so that it gets self-stimulation when it binds to the target so that this CAR T cell will kill it. So we talked about CD8, cytotoxic T cell, your cold killer assassins. Now CD4 T helper cells, the instructors, the teachers, if you will. These T helper cells have CD4 antigens, and they are subdivided to Th0, Th1, Th2, Th17. The Th0 helper cells are generated in the thymus. Once they leave the thymus, they become Th1, Th2, or Th17 helper cells. So when they leave the thymus, how do they become these different Th1, Th2, Th17 T cells? And they do that by post-thymic cytokine activation of a master transcription factor. A single master transcription factor will flip a Th0 cell from the thymus into a Th1, 2, or 17 cell. So the T helper cell, which can help activate a cytotoxic T lymphocytes, that master transcription factor is called T-BET. So a Th2 cell that activates B lymphocytes or plasma cells to make antibodies, that master transcription factor is called GATA3. T helper cell, that is the Th17, that activates inflammatory immunity at barrier surfaces such as your gut, is ROR. And the T helper cell called Tregs that shut off inflammation, that master transcription factor is FOXP3. All right, so this is kind of a brief summary of how that works. So you see in the thymus on the left side of the screen, you have in the thymus antigen presenting cells that present to T cells through the T cell receptor. And in this case, it's a CD4 because it's going to become a CD4 helper cell. And the co-stimulatory molecule called CD80 on the antigen presenting cell and CD28 on the T cell. Now, it sees this peptide, it gets a co-stimulation, and now it goes into the periphery. And in the periphery, it's a CD4 positive Th0 cell. And out in the periphery, depending upon the cytokine that it interacts with, it can become if it's IL-4 on the right, a Th2 cell that helps B cells make antibodies or instructs them to do it. Or it can become a Th1 cell to help cytotoxic T lymphocytes with the right cytokines. Also, with the right cytokines, it can become a Th17 cell at mucosal barriers for immunity, or it can become an induced Treg cell under the right cytokine influence. And remember, these Treg cells, CD4 in this case, CD25 FOXP3, can come directly from the thymus as natural Tregs or as induced Tregs from the periphery Th0 cell. This is all great, except the problem is, again, the immune system is a system of balance. It's kind of a system of diplomacy, if you will, finding the right balance between parties, because these Th cells, once they're made, they're not set in stone. They have plasticity. They can flip. A Treg cell can become a Th17 or a Th1 cell, for instance. So they can change their function after that master transcription factor is turned on. It can be shut off and another master transcription factor activated. So that's a problem for using Treg cells to treat autoimmune disease, because in the periphery, they can switch to T effector cells. And so instead of stopping the disease, they can contribute to it. But also another problem when we try to use Treg cells alone to treat autoimmune disease is they're not antigen specific. They're just broad Tregs with all kinds of different T cell receptors in them. So whereas what you want to fight a particular infection or to stop it or downregulate your effector T cells from causing that infection is a Treg cell that's specific for a particular self-epitope. Okay, how infections, viruses, and vaccines stimulate autoimmune disease. So again, your innate immune system doesn't attack self, but adaptive will if it gets misdirected. How does that happen? It happens through a concept called molecular mimicry. In other words, the pathogens, that antigen or epitope on the pathogen that it recognizes, that your adaptive T cell recognizes, shares the same or similar amino acid sequence to a self-epitope. And so you get an autoimmune disease. And that's documented in the literature. Guillain-Barré from Campylobacter jejuni. Heliobacter pylori can cause ITP. Once your immune system attacks Heliobacter, can cross over because of molecular mimicry to platelets, give ITP, Streptococcus rheumatic fever, Epstein-Barr virus disease, multiple sclerosis, and Coxsackie virus B, type 1 diabetes mellitus. And kids, before type 1 diabetes show up, not unusually will have a Coxsackie virus. The immune system, the adaptive, gets activated to help fight it, clean it up, and that it can cross over and attack beta cells and give through molecular mimicry type 1 diabetes. And it can do it by epitope spreading, which means the immune response starts against a dominant epitope and then spreads to other epitope in the same other proteins. And that's been well documented, especially in animal autoimmune diseases, where you initiate it against a small 8 amino, 10 amino acid sequence of a peptide, and then it crosses. Once it starts, the immune system becomes activated to other peptides in that protein, as well as other proteins in that tissue. And that's happens to multiple sclerosis, pemphigus vulgaris, type 1 diabetes, probably others, but it's been documented there. And it can be a bystander effect. In other words, non-specific activation of your immune cells by inflammation and cytokines and destruction of a pathogen or the cell infected with the pathogen can cause tissue damage, which then can cause the antigen-presenting cells, the dendritic cells, to present self-damaged tissue with co-stimulation to your T cells, which then cause your T cells to attack self. And that's happens in rheumatoid arthritis, multiple sclerosis, celiac disease, and SLE. So vaccines are viruses, either live attenuated viruses, a dead virus that's has an intact 3D structure so the immune system can recognize it and attack it, or a piece of a virus, such as just the spike protein on the virus, or the messenger RNA that makes a piece of the virus. And vaccines, like viruses, activate innate immunity that activates the adaptive CD8 and CD killer cells and CD4 effector cells that may lead to autoimmune disease in animals by one of the mechanisms we just talked about: molecular mimicry, epitope spreading, and bystander effect. How do we know this happens? We know with the Pasteur vaccine developed in the early 1900s in Paris by Louis Pasteur. And what he found is that when he, infected rabbits with the rabies virus and took out their spinal cord and then either embalmed it or air dried it for 6, 7 days, and then injected that uh, into a person, who had been bitten by a rabid animal, you could prevent them from getting rabies. Unfortunately, about 10% of the patients also developed an MS-like disease, which was called experimental allergic encephalomyelitis. But then that was because it occurred in humans, they had to figure out how to prevent that. Now, rabies is 100% lethal once you have symptoms. So you've converted 100% lethal disease to about 5 to 10% of patients having an aggressive MS type of disease. But what they found is it was the myelin, the protein around neurons in the spinal cord, doing it. Once they removed that, they got rid of vaccine-induced EAE, an MS-like disease. But because that was discovered as kind of a surprise in patients receiving the Pasteur vaccine who had been bitten by a rabid animal, it was realized we could take that concept of to induce autoimmune diseases in animals and then study the autoimmune disease. And that concept is to use the vaccine as an adjuvant to stimulate your innate immune system. And besides using that vaccine or that viral epitope as an adjuvant, you add a self-peptide. So the innate immune system will attack the vaccine or the adjuvant, but then cross over by one of the mechanisms we talked about, molecular mimicry, epitope spitting, or bystander effect, to attack the self-peptide to start an autoimmune disease. And so in the lab, immunologists do that all the time to make autoimmune diseases in animals and then to study it. So, of course, experimental autoimmune encephalitis, that's an animal model for MS. It's done by using a little peptide of proteolipid protein, which is found only in the proteins of myelin in the central nervous system, not the peripheral nervous system. Collagen or adjuvant-induced arthritis is another example. There's a whole variety of examples. Experimental autoimmune myasthenia gravis, which is myasthenia gravis, by using an adjuvant that is something that stimulates innate immunity along with the acetylcholine receptor peptide or a piece of it. And then you can just use an adjuvant itself to do it, whether it's adjuvant arthritis or adjuvant-induced lupus. Activating the innate immune system, because it can damage local tissue, get a bystander effect, can cause the autoimmune disorders in animals. What about humans? So it's done all the time in animals for a whole variety of autoimmune diseases, but in humans, epidemiologic studies do not in general support an association between vaccines and autoimmune diseases. There are exceptions. In the general public, the influenza vaccine can cause Guillain-Barré syndrome, and the measles, mumps, rubella vaccine can cause idiopathic thrombocytic purpura, or ITP. The COVID-19 vaccine can cause ITP. And there are now some publications that in genetically predisposed individuals, the COVID-19 vaccine can cause multiple sclerosis. And I put that with a question, but there are now publications out there saying that. Genetically predisposed means like a family history or someone who had MS and was put in a complete remission by hematopoietic stem cell transplant, but then get the COVID vaccine and relapses. So it's important to know that epidemiology is very, very, very powerful tool. But what it does is it draws associations. It implies guilt or innocence because of those associations, but it does not prove guilt or innocence. That's really important to understand that. How are autoimmune diseases treated? Traditionally, you use chronic immune suppressive drugs to just tamper it down, but the patients are on it long term. Sometimes they get off and stay in remission because there's enough of a reset when the inflammation stops of Treg cells coming in that you can get drug-free remission. But oftentimes you're just stuck on these immune suppressive drugs that are changed, and you have all the side effects from it. Or you can do a reset of one immune compartment, such as B cells. And they're doing that right now in lupus using chimeric antigen receptor T cells directed against CD20 on the B cell to reset, knock out the B cell and reset the B cell compartment. Or you can do an immune reset of both T and B cells. And that's what hematopoietic stem cell transplant does. And that's far more powerful because T cells do not act in isolation. B cells do not act in isolation. T and B cells interact with the innate immune system, and T and B cells interact with each other. I just didn't show that because there's not enough time. They don't act in isolation. And so when you immune reset both, it's a much more powerful way of inducing a very long-term remission of an autoimmune disorder. And that's the rationale between HSCT for autoimmune diseases that I pioneered. All right, so HSCT for autoimmune diseases resets the adaptive immune system. It transiently removes a large number of these effector T cells and B cells. And then there's a surge in Tregs that prevent inflammation, and it reestablishes tolerance. So you have your innate immune system. It's non-specific for immunity, but towards pathogens, but it doesn't cause self-autoimmune diseases. Well, almost never. And then you have your adaptive immune system, which is your cavalry to come in, protect you from a specific infection. It has to learn how to do that. But if things go awry, it can cause an autoimmune disease. And HSCT resets the adaptive immune system. It markedly lowers the effector pathogenic cells and upregulates the Treg cells, while new naive effector cells are being regenerated. So I have a book on this for everyday people that don't have a medical background, but as a physician, I think you'll really enjoy it. It's like chicken soup or kitchen soup for the soul. And you can get it wherever you buy books. And the forward's actually done by the Dalai Lama. His quote in the forward is there on this slide. You will really enjoy reading it, or if you're a patient, you will learn a lot. And the basic rationale for how I came to do this and why it works, I just presented. I also did another lay book, and it's very important given what's recently happened with COVID-19. It's about activating inflammation with vaccines. That book is called Kill Switch, The History of How Viruses Shaped Humanity and Led to COVID-19. Again, I read it for people without a medical background, but anybody in medicine is going to learn a tremendous amount from this. We have an ecclesiastical forward by the Vatican, Monsieur Trafny, the head of culture and ethics at the Vatican, and a secular forward by the Honorable U.S. Senator from the state of Illinois, Mark Kirk. Thank you.